Welcome to Future Talk. This is the show that examines the global impact of technology and tries to see where it's leading us. Technology is changing the world at an extremely rapid rate, but have we really thought through the implications of these changes? Technology contains a lot of promise, but it also contains some perils. On Future Talk, we examine both the promise and the perils. On today's program, we're going to talk about a topic that's very much in the news, human genome research. We have three guests to cover it from three different angles. Gavin Sherlock is an assistant professor of genetics at Stanford Medical School. He'll discuss the latest scientific developments in genome research. Jack Porter is a Silicon Valley entrepreneur who's helped start several pharmaceutical companies. He's currently president and CEO of a company called Forward Innovations. He'll talk about the practical applications of genomics. And Mark Feldman, professor of biology and co-founder of the Human Genome Diversity Project at Stanford, will talk about the moral and ethical implications of genome research. We're going to start with Gavin Sherlock, but first, we have a short video that gives an introduction to genome science. Let's take a look.
If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life.
That was a brief introduction to genome science, and I'm talking with Gavin Sherlock. Gavin, what would you say is the most promising area in genome research right now?
Well, at the moment, research is really being driven by advances in technology. So the ability to sequence DNA is driving research forward at an incredible pace. So for example, the Human Genome Project, to sequence those 3 billion bases, took about a decade and took hundreds of scientists to do so. Now you can do a human genome for maybe $50,000 in a few months. And it's increasing probably so that, maybe in a year's time or 5 years' time, maybe it's going to be $1,000 for a human genome.
So what are some of the interesting things that we've found out about the human genome since we've been doing this research?
So there's a number of things that we've been able to find out. For example, one thing that was a big surprise to people is that there are genes which are present in some people, absent in other people, or maybe present in more than one copy in some people. Another project that's been receiving a lot of attention recently is something called the TCGA project, or Cancer Genome Atlas project. And what they've been doing is examining hundreds of cancer samples to identify genes which, for instance, might be turned on in cancer or turned off in cancer or amplified in cancer. And they've been really able to understand what pathways are changing in cancer, for example, in brain cancer or ovarian cancer. And the hope is that that's going to lead to either better diagnostics or better prognostics, so we might know how to better treat people with those diseases.
One thing that I've always been interested in is, does the genome code for intangible traits such as intelligence or personality, or is it all just physical characteristics?
It does encode for those intangible traits, at least to some extent. I mean, what we are is eventually a product of both our genes and our environment. And for many of these very difficult to understand traits, such as either intelligence or maybe something like schizophrenia, it's not just one gene, it's many, possibly dozens or even hundreds of genes that all each have a small influence. So yes, children might, who have smart parents might be more intelligent on average, but it's no means a certainty. It depends what their environment might be.
Is there such a thing as a gene that by itself increases the chances that you'll have some disease? Or, I mean, is it like a negative trait, or is it just a variant that gives some kind of weakness in a certain area?
For the most part, I think genes which contribute to disease are just natural variants of genes that normally carry out a job in the genome. They may be a mutation, or they may be a variant which, in most people, maybe it doesn't even make a difference, but the interaction with the environment might cause it to have an effect, such as lung cancer and smoking. If you didn't smoke, maybe you wouldn't get lung cancer, even though your genetics might predispose you to it.
Is there anything that scientists are really looking forward to discovering that they think they're pretty close to?
Well, I would say the things that we're looking forward to discovering are the things that we can't even imagine yet. For example, something called RNA interference. We didn't really know anything about that 15 years ago. We didn't even know it existed. But now we know that there's a whole network that cells use to regulate how their messages are turned into proteins. And I suspect there's lots more surprises like that out there for us.
We're going to move on to our next segment, which is talking about the practical applications of genome research. But first, we have a short video that demonstrates one such application. Let's take a look.